Here I am starting with the graphs of the experimental lab which are asked frequently and viva questions related to that. So first graph which we can get in the experimental exam is simple muscle twitch or simple muscle curve. So something like this kind of graph you may obtain in the exam, and you will be asked. The first most important question will be identify the graph. So identification of the graph is most important thing to be asked. So how will you identify the graph? There will be single graph and there will be different phases of that graph. So this is simple muscle twitch or simple muscle curve. So how do we obtain the simple muscle curve? We usually take the nerve muscle preparation of any amphibian, mostly that is frog. So what is that nerve? That nerve is sciatic nerve and muscle is gastrocnemius muscle. So we dissect the frog and we obtain these muscle nerve preparation, and then we put on the myograph board, and then we are going to obtain the graph. So this is the simple graph which is obtained by giving stimulus at the sciatic nerve, and due to which the impulse will be transmitted through the nerve and then through the muscle. Muscle will contract because of that, and that contraction we trace on the drum. So that kind of graph will be obtained. Now we must know the different phases of the graph. So this is at this point, this is point of stimulation. So at this point we are going to give the stimulus, and then after some time there will be gap of some time, and then we are going to obtain the graph like this. So this is the point of maximum contraction, and this is the point of complete relaxation. This is the point of initiation of the contraction. So this is point of stimulation, this is point of initiation of the contraction, this is point of the maximum contraction, and then relaxation starts, and this is the point of complete relaxation. So there are different phases we can see. This is latent phase. So latent phase is from point of stimulation to the point of start of contraction, latent phase, during which there will not be any activity, will not be visible. Now there is starting of contraction, and contraction peaks at this point, and then there is relaxation. So contraction phase is from the point of initiation of the contraction till the maximum contraction. Relaxation phase is from the point of maximum contraction till the relaxation is complete. Then there are some smaller curves we can see. So these smaller curves are called as physiological curves. So examiner may ask you what is the cause of these physiological curves. So these physiological curves are not a physiological phenomena. So this is misnomer. These are not physiological. These are because of inertia of the liver. Liver means the instrument which we used in the practical. So there is some vibration after the relaxation is over. There is some vibration in that instrument. Because of that, these physiological curves are formed. So this is misnomer, these are not physiological. This is because of inertia of the liver. Here we can see. So now we must know what is the normal duration of these curves, these phases in case of frog. So normally the duration of the latent phase is 0.01 second. Duration of the contraction phase is 0.04 seconds. Duration of the relaxation phase is 0.05 seconds. So this is approximate duration of all these phases. Okay, what may be the next question of your examiner, that how would you measure the different phases? So this measurement is done with the help of time tracer. So along with tracing the graph, we also put time tracer on the drum. So because of that time tracer, these small curves are formed. So duration of these curves is dependent upon the frequency of that particular time tracer. So mostly the time tracer which we use in this experiment has 100 Hz frequency. 100 Hz frequency means the single duration of the single curve is about 1 by 100, means 0.01 seconds. 100 Hz means 100 rotations, 100 vibrations per second, means duration of the one curve is equal to 1 by 100 or 0.01 seconds. So that is how we can measure the duration of different phases. Like for latent phase we can count this complete curve is complete. So the duration of latent phase is 0.01 seconds. If duration of the contraction phase is calculated, then look at this, how many curves are complete? 1, 2, 3, 4. 4 curves are complete. So duration of contraction phase is 0.04 seconds. Duration of relaxation phase 1, 2, 3, 4, 5. So about 5 curves are completed. So duration is 0.05 seconds. So this may vary in your graphs. Okay, this may vary little bit, like duration of the relaxation phase may be 0.055 seconds. So this may vary little bit. Now the next question which your examiner can ask is, tell me the causes of latent phase. Causes of latent phase, because stimulus is given at this point and why the contraction has been started after some time. So from where this time is coming. So let us see how we are going to give the stimulus. This is muscle. This is, suppose this is gastrocnemius muscle and this is nerve which is attached to it. This is tendon of the muscle. This is isotonic muscle liver which is making graph on this drum. Okay, this is drum. So we are giving a stimulus at this point. So stimulus will first travel from this point to this point through the nerve, and then it will pass through the neuromuscular junction, that it will reach the muscle, there will be excitation, contraction, coupling, and that stimulus, there will be contraction of muscle, and then because of that contraction of muscle, this liver, because there is inertia in all the mechanical instruments, there is some inertia. So this isotonic muscle liver will take some time to raise. Okay, so that time is also included in that. So what are the causes of latent phase? Let us see. First, the time taken by impulse to travel from point of stimulation till the neuromuscular junction. Then time taken at the neuromuscular junction, means the event which are taking at the neuromuscular junction, like release of neurotransmitter, entry of sodium ions and all that. And then time taken in the muscle, where, because there is some viscosity in the muscles also. So actin and myosin fibers will feel kind of resistance in that, and also excitation and contraction coupling of the actin myosin will take some time. So that time is there, and inertia of the liver, inertia of the mechanical instrument is also taking some time. So these are all causes of latent phase. So point 1 is time taken by traveling of the impulse from point of stimulation till the neuromuscular junction, time taken at the neuromuscular junction, time taken in the excitation, contraction, coupling, then time taken by the inertia of the liver. So these are all causes of latent phase. So this is very common question which is being asked. So this is about the simple muscle curve and common viva questions which are asked in simple muscle curve. So next graph you can get in your exams is effect of temperature on simple muscle curve or simple muscle twitch. So in that you are going to get 3 graphs like this. Okay, so here temperature is written over there. So this was the graph on normal temperature. This green one is at cold temperature and this one is at warm temperature. So what we do to produce that effect, we pour a cold saline or warm saline on the nerve muscle preparation and then we obtain the graph. So we can see this is the normal, this is the graph at normal temperature. When we poured warm saline on this, so we can see the height of the curve is increasing and duration of all the phases are decreasing, like latent phase, contraction phase, relaxation phase. So duration of all the phases have decreased and length of the curve or amplitude of the curve has increased. Or in other words, we can say the force of contraction has increased or the velocity of contraction has increased. In cold temperature, we can see the duration of all the phases have increased, but amplitude of the contraction has decreased. Or in other words, we can say force of the contraction has decreased or velocity of the contraction have decreased. So here we can write down that what has happened on pouring warm saline, amplitude of the contraction has increased, latent phase, contraction phase and relaxation phase duration has been decreased. On pouring cold saline, amplitude has decreased, latent phase, contraction phase and relaxation phase have increased. Duration of these all three phases have been increased. It means the contraction have become slow. Now another question your examiner may ask, that why this effect has occurred. So what happens on pouring warm saline? So whenever temperature increases around the muscle, so what happens? Temperature of the muscles have also increased. So because of that, enzymatic activity will increase. So on increasing enzymatic activities, all the process have been fastened. Okay. What else? On increasing temperature, viscosity of the media like actin and myosin are contracting. So viscosity around them decreases. So this viscosity was creating a kind of resistance for them. So resistance have decreased. So because of that, contraction of the actin and myosin have become faster. So because of increased temperature, there is increasing enzymatic activity, there is decreased viscosity, and because of that there is decreased resistance around the actin and myosin. So now the actin and myosin will contract more vigorously, okay, will contract more fast. So this has happened because of this, and opposite will occur in pouring cold saline. There is more term heat rigor. So what happens in heat rigor? In heat rigor, in place of pouring warm saline, in case if we are pouring hot saline, what happens? The muscle protein will get denatured, muscle protein will get destroyed, so muscle will lose its property of contraction. So we will not be able to study the properties of muscles because we have already poured the hot saline. So this condition is called as heat rigor. So similar to that, like name is very much similar to that. So examiner may ask you that also, what is rigor mortis? Although rigor mortis has nothing to do with the rigor, heat rigor, but examiner may ask you, what is rigor mortis? Rigor mortis is a state of permanent contraction. Okay, there is permanent contraction without relaxation. Why? Because for relaxation of the muscles, ATPs are required. So here we don't have ATPs after death. So few hours after death, like 15 to 20 hours, maybe 24 hours, that will depend upon many factors, that what happens, muscles will go into permanent contraction, there will be no relaxation and muscles will become rigid. So this rigidity of the muscles is called as rigor mortis. Now this rigidity is dissolved after some time. Why? Probably because of release of lysosomal enzymes which will cause the autolysis. So this rigidity will be lost after few hours, and this is how the rigor mortis will be resolved. So this is what is rigor mortis, and this is the cause for dissolution of rigor mortis. So these are all questions your examiner may ask. The causes, what is the most important question? Identify the graph. Then why does this occur? What is the cause for the raising of the graph, and what is the cause for changing of the duration different phases? And then he may ask you what is heat rigor, he may ask you rigor mortis. So this is all about the effect of temperature on simple muscle curve. So the next graph you can get in your exams is effect of repeated stimuli on simple muscle curve or simple muscle twitch, that is genesis of fatigue. So in that you will get multiple curves like this that will be of different height, and the baseline will be shifted gradually upwards like this. Okay, so first few curves you can see the height is increasing, but then height is gradually decreasing. So why? On giving repeated stimuli of same strength, height of the curves are increasing. That is because of beneficial effect. But then you can see what happens, the height is gradually decreasing, and also the duration of all the phases are increasing gradually, and then later on the baseline is also coming little upwards. Okay, now this phenomena of baseline to come upwards is called as contraction remainder. Now the first and most important question in all the graphs may be identify the graph. So you would see the graph, there are multiple graphs of different heights and baseline is shifted upwards. So that is a graph of effect of repeated stimuli on simple muscle curve or genesis of fatigue. Now the second question may be what is fatigue? So what is fatigue? Fatigue is the decreasing working capacity of the muscle or cell on repeated contraction. So what is the cause of fatigue? The cause of fatigue may be on repeated stimuli, on repeated several stimuli or on repeated contraction. There may be decreased nutrient and oxygen supply. There may be depletion of acetylcholine stores and there may be accumulation of the metabolites. So all these may be causes of fatigue. Now the next question may be fatigue is reversible or irreversible. So fatigue is a reversible phenomena. On giving some rest, the fatigue weans off. Why? Because on giving some rest, there is supply of oxygen, there is supply of nutrients, acetylcholine will get stored in the vesicles, and of course there will be removal of the stored metabolites like lactic acid, potassium ions and all that. So fatigue is a reversible phenomenon. Next question may be what is the cause for increased height of curves in the beginning. In the beginning we can see there is for few curves there is increased height of the curves. So why is that? That is because of beneficial effect. Now what is beneficial effect? In beneficial effect there is increased force of contraction, or in other words there is increased height of contraction for few contractions. Why? Because gradually later on contractions are getting benefit from the previous contraction. Now what is benefit from the previous contraction? In previous contraction there is release of calcium ions. So that calcium ions will take some time to get entry into the sarcoplasmic reticulum, because calcium ion come rapidly out of that but go little slowly into the sarcoplasmic reticulum. So more and more calcium ion will get accumulate with repeated contraction. So now because more calcium ions are available, now because of that there will be more forceful contraction of the muscle, because of more number of actin myosin fibers will get involved. Next, because of repeated contraction there is increased temperature, there is increased enzymatic activity, there is increased metabolic activity in the cell. So that will also do forceful contraction or increased height of contraction. Next cause may be because of increased temperature there is decreased viscosity around the muscle fibers. So because of that there will be decreased resistance around the muscle or inside the muscle. So now the actin myosin fibers will contract more vigorously, more forcefully. So that is also cause for the increased force of contraction, or in other words beneficial effects. So one is more release of calcium ions, more temperature, more enzymatic activity, more metabolic activity. More temperature will lead to decreased resistance, decreased viscosity, decreased resistance. So that will lead to increase of forceful contraction of the actin and myosin filaments. Next question may be why baseline is shifted upwards with repeated contraction. Why baseline comes up? So this phenomena is also known as contraction remainder. So with repeated contraction when the fatigue sets in, the muscle is not able to relax completely. So here there is incomplete relaxation of the muscle. Why there is incomplete relaxation? Because we know that for relaxation we need ATPs. For detachment of the actin and myosin we need ATPs. Now because during fatigue there is decreased supply of the ATPs, so that will lead to incomplete relaxation of the muscle, that will lead to the contraction remainder or upward shifting of the baseline of the curves. So this is about fatigue, beneficial effect and contraction remainder. Next question may be what is the site of fatigue in a nerve muscle preparation? So in nerve muscle preparation there is a nerve, there is neuromuscular junction and there is a muscle. So what is site of fatigue? Where the fatigue sets earlier? So that is neuromuscular junction, where the fatigue sets earlier. If we talk about intact human beings, so fatigue sets earlier in CNS. and that in the synapses of the brain. So synapses of the brain is a site where fatigue sets earlier. So this is all about this curve and the important question which are asked in the Viva. So now we are starting the other experimental graph, that is effect of increasing strength of stimuli on simple muscle curve. So in that you are going to get graphs like this. So looking at the graphs like this, you would have a few things in your mind, that why like other practicals you are not getting curve like this. Like you have seen, you are getting curves like this in other practicals. Why not in this practical? See, suppose if we are doing, we are making graph on any curved surface, okay, and that curved surface is suppose moving, then only we can get graph like this. So these graphs we are making on curved surface, but that is stationary. So we are doing this practical on stationary drum. Suppose if we will draw perpendicular on this, it will be like this. On any curved surface, if we will try to draw perpendicular, so it will be drawn like this. So in any practical, if we don't need different time of different phases, like these phases, so that curve we can make on stationary surface. So these curves are made on stationary drum. So that is why we are getting graphs like this. There's one thing, that we have shifted the drum little bit. Every time we have shifted the drum little bit, so to get another graph. So that is why we have obtained graph like this. Second thing, that you must be thinking what are these numbers, 20, 19, 18, 17, 16. As the number is decreasing, length of the graph is increasing. So what are these numbers? These numbers actually show you the distance between two coils. So while these graphs were made, actually we were using induced current which were produced in the secondary coil. So and that induced current was produced because of change in flux in the primary coil. So as we were decreasing the distance between two coils, there was increased strength of stimuli in the there was increased current in the secondary coil. So that is why there is increased strength of stimulus gradually. So as we are decreasing the distance between two coils, there is increased current in the secondary coil. So that is why these numbers are being written. Third thing which may come into your mind is why MB? What is this MB, MB, MB? So meaning of this MB is make shock and break shock. So we know that induced current is produced only when there is any change in the flux in the primary circuit. So when the current was starting means make shock, or the current was breaking means break shock. Only that time there was production of induced current in the secondary circuit. So that is why it is showing make shock or break shock. At a single point there may be two strength of currents, one at the make, one at the break. So that is why the meaning is make and break. Now what happens in that? We can see in this graph there is increased length of curves, increasing length of curves. So what is meaning of that? See, we are gradually, suppose this is one motor unit. This is one muscle, and suppose there are 3 motor units in that. Although in any muscle there are multiple motor units, but here for understanding I am drawing 3 motor units. One has threshold of 5, another has threshold of 10, and third one has threshold of 15. So suppose we are giving current of different strengths. So in the beginning, suppose I have given a current of stimulus of 2 millivolts. Then will there be any response? No, because it is below threshold. But then I gave a current of 5 millivolts. So this one of motor units has threshold of 5. So this one motor unit will contract, and then muscle will also contract. If again we are giving a stimulus of 10 millivolts, what happens? Now this motor unit and this motor unit will come in picture because now this is crossing the threshold of these two motor units, and now the contraction of the muscle will be little more stronger. Now again, if I am giving a stimulus of 12 millivolts, will there be any change in the force of contraction of muscle? No, because previously also two motor units were there, and now also two motor units will be there to contract the muscle. Now again, suppose I have given a stimulus of 15 millivolts. Now we will see that all the three motor units will come in picture because now my stimulus has crossed the threshold of all the three motor units, and now the muscle will contract to its maximum. Again, if I have given a stimulus of 20 millivolts, will there be any change in the force of contraction? No, because all the three motor units have come in picture. So gradually, as we have increased strength of stimulus, more and more motor units kept on adding, and more stronger will be the force of contraction of muscle. So this phenomena is called as recruitment phenomena. So what is recruitment phenomena? As we keep on increasing strength of stimuli, more and more motor units will keep on adding, and more stronger will be the force of contraction of muscle. So by this example we can understand here. So here we have given a stimulus, here a stimulus, so there was no response. At this point, at the 18 centimeter distance of between two coils, there was some response, which is called as threshold stimulus. So here we can understand for this muscle it was 5 millivolts. Okay, so at the point when at least one motor unit will come in picture, so that is the threshold stimulus. But then we kept on increasing the force, the strength of stimuli, the force of contraction kept on increasing. At one point it was maximum. Okay, after that we kept on increasing the strength of stimuli, but there was no change in the force of contraction of muscle. Why? Because already all the motor units have been recruited, have been added. So the examiner may ask you the definition. What is this? The minimum stimulus at which at least some response was produced is called as minimal or threshold or liminal. It is liminal, not luminal. It is liminal stimulus. So what is threshold stimulus? Minimum stimulus at which the response was produced. At least some response was produced is the threshold or minimal stimuli. Now, what is maximal stimulus? Maximal stimulus is the minimum stimulus at which the maximum response is achieved. We can see the maximum response is achieved in all four graphs, but this was the minimal stimulus at which the maximum response was achieved, is maximal stimulus. Okay. What is submaximal? The responses between the threshold stimulus and maximal are the submaximal, and the responses after maximal stimuli are supramaximal stimuli. Okay, so here these are supramaximal stimuli. We kept on increasing the strength of stimuli, but there was no change in the height of curves, or in other words, there was no change in the force of contraction of muscle. Okay, so these are few definitions here. Now your examiner may ask you that why here all or none law is not followed. Okay, we have seen that on increasing strength, force of contraction kept on increasing. So we can answer that all or none law is true for one motor unit, but here we have multiple motor units. Okay, so here all or none law does not apply. Then now your examiner may ask you what is all or none law. So all or none law is within same environmental conditions. If a stimulus is being given to a motor unit, that motor unit will contract to its maximum or not at all. Suppose this stimulus is more than threshold, equal to or more than threshold stimulus, the motor unit will contract to its maximum, or if it is less than threshold, it will not contract at all. This is all or none law. All or none law is true for muscle fibers, nerve fibers, motor units and heart as a whole, but not for muscle as a whole. okay, but here we are taking muscle, not single motor unit. Now your examiner may ask you what is motor unit, then you must know motor unit is single motor neuron and muscle fibers supplied by it constitute one motor unit. So this is all about these graphs. Many times when make and break shock both are not used, you may get single graph, okay. In many experiments arrangements are done in such a way that they use only break shocks. In those experiments you will not get two curves at each distance, you will get only one curve that is made with the break shock only. Many time you should not get confused, many time you will get straight lines like this, okay. So why we are getting curved line like this, curved, because we are making these graphs on the cylindrical surfaces, so we will get curved lines. But when we are getting straight lines, it means it is made on plane surface. When these are made on plane surface, we will get straight lines. So in few cases you may get straight lines like this. Now very rarely where the these experiments are taught in detail, they may ask you why break shock is stronger than the make shock. You can see that the lines of the break shock is little higher than the make shock. So why that occurs? Then some books have given explanation that in break shock the current in the primary and secondary coils are in same direction, and in make shock they are in opposite direction. So current produced during break shock is little more than the make shock. So this is all about these graphs. So I will revise the important Viva questions. Identify the graph. This is the most important question. Identify the graph, and what are these threshold stimulus, what is maximal stimulus, what is supramaximal, what is submaximal stimuli, and then you must know what is recruitment phenomena, you must know all or none law, you must know motor units. So all these may be asked for your Viva. So that was all about this curve, this graph, and important Viva questions related to it.